Good morning. This is the lecture for Friday, October 30th, 2020. This will cover the introduction, the first half of Chapter 14. Chapter 14 deals with carbohydrates. Carbohydrates are most often or most frequently known as sugars or starches. More technical names for these are polyhydroxylated aldehydes or ketones, or compounds that can be reacted with water to produce them. Carbohydrates can be further subdivided to monosaccharides, disaccharides, and polysaccharides. Most of the time, these mono, di, and polysaccharides are made in green plants through the process of photosynthesis. This is the processes that use the energy from the sun to convert carbon dioxide and water into glucose and oxygen. In the body, these carbohydrates are used for energy needed during exercise in the form of glucose, which is the fundamental energy source in the human body. So we have this energy cycle. The plant takes CO2 and water, converts it via the process of absorbing a photon of light and the enzyme chlorophyll to create glucose. The bond energy that's stored in the molecule of glucose is released during the process of human metabolism to produce CO2, water, and energy. And the process starts all over again. First, simplest kind of saccharides are known as monosaccharides. These are the simplest of your carbohydrates. They have generally 3 to 6 carbon atoms in a chain with a carbonyl group at C1 or C2 and OH groups, depending on the identity, at C3, 4, 5, and 6. Of the monosaccharides, they can be further classified as either those that have the carbonyl at C1, and those are known as aldoses, or the carbonyl at C2. These are known as ketoses. So, monosaccharides, generally 3 to 6 carbons. At C1 or C2, you have your C double bond O. If you have the carbonyl at C1, it's an aldehyde, given the designation an aldose. The carbonyl is at C2, it is a ketone, known as a ketose. C3, 4, 5, and 6 have either one or more than one OH group attached to them. The simplest aldose, 3 carbons, is glyceraldehyde. So at C1, you have your C double bond O, hydrogen. C2, C3. Both C2 and C3 have OH groups. Since the carbonyl is at C1, it's an aldehyde. It's an aldehyde, so it's aldose. The simplest ketose is dihydroxyacetone. Since it's a ketose, we have a C double bond O attached to 2 different carbon groups. C1, here we have the C double bond at C2. The other carbons have OH groups on them. So the simplest aldose is glyceraldehyde, 3 carbon. The simplest ketose is this 3 carbon system, dihydroxyacetone. Both of these are constitutional isomers of each other. They both have the formula C3H6O3. So 1, 2, 3 carbons, 1, 2, 3 oxygens, 1, 2, 3, 4, 5, 6 hydrogens. Again, 3 carbons, 1, 2, 3, 4, 5, 6 hydrogens, 1, 2, 3 oxygens. So both of these are constitutional isomers of one another. We can further distinguish monosaccharides by the numbers of carbon atoms in their respective chains. A triose has 3 carbon atoms in its chain. A tetrose has 4 carbon atoms in the chain. A pentose has 5 carbon atoms, and a hexose has 6 carbon atoms. We can then combine the triose, the pentose, the tetrose, the hexose with the aldose and ketose designations to further subclassify monosaccharides. So glyceraldehyde is an aldotriose. Glyceraldehyde, 3 carbons in the monosaccharide chain. That makes it triose. But since the C double bond O is at the first carbon, it is an aldotriose. Dihydroxyacetone is a ketotriose. Keto, carbon is at C2. Triose, 3 carbons in that sequence. So any combination of 3, 4, 5, and 6 with aldose and keose. So you can have an aldopentose. You can have a ketotetrose, an aldohexose, a ketohexose. Again, triose, pentose, triose, tetrose, pentose, hexose deals with the number of carbon atoms. The aldoketo deals with where the C double bond O is at. If it's at C1, it's an aldo. If it's at C2, it's a keto. If we deal with stereochemistry, again, the positions of the groups about particular carbon atoms can create chiral centers. So all carbohydrates except dihydroxyacetone have 1 or more chirality centers. Glyceraldehyde is the simplest aldose and has 1 center of chirality. Therefore, it has 2 possible enantiomers. Here's this chirality center here. It's attached to 4 different things: hydrogen, the aldehyde, the OH, CH2OH. This carbon is a chiral center, again, attached to 4 different things, but the 3-dimensional positioning of the OH here and in its mirror images are different from one another. If we look at the 3-dimensional presentation of these around this chiral center, here is our 1 position, because this is the group of most highest priority. This guy, then that guy. So we're going counterclockwise around this direction. Here we're starting with priority here, 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 then there. So we're looking at a clockwise orientation. These are 2 different compounds. They are related to one another as enantiomers, but they are different compounds. This is the naturally occurring isomer. The body generally produces, when we have stereocenters, the body is unique that it only produces one particular isomer and not the other. So here we have the unnatural isomer, L-glyceraldehyde. When we're dealing with Fischer projections, the prefix D is used when the OH group is drawn on the right side of the carbon chain. So here is our carbon chain. We see the OH group here referred to as the D isomer, because this OH is written to the right of that carbon chain. Here, this is the L isomer because this OH group is to the left of your carbon chain. So we can redraw from these Fischer, from these Fischer projections back into the wedged and dashed lines can be redrawn into a Fisher projection. So the OH is on the right, it ends up on the right here. The hydrogen is to the left, CHO is up, CH2OH is down. What happens now? This is all fine and well and good if you only have one chiral center. What if you have more than one chiral center? If we look at glucose, it has 4 chiral centers. 1, 2, 3, 4 chiral centers. We can represent that molecule glucose on the far right, on the far left, with its Fisher projection on the far right. Wherever we have a chiral carbon, we write a cross. So there's your vertical, there's your horizontal. The position of these is the same as it is here. So since this OH is to the right of the vertical carbon chain, this OH is to the right of it. Since this OH on the next carbon down is to the left of the carbon chain, it's to the left. This OH is to the right, this OH is to the right. How do you use D and L when you have multiple chiral centers? You can use the D and L by defining where the OH is on this last OH group. That is the farthest chiral center from your carbonyl group. This carbon is not chiral. It has only 3 different things attached to it. 2 of the things are identical and they're both hydrogen. So this is the last carbon that is chiral from the farthest end. That determines whether this particular compound is referred to as D or L. Since this OH is on the right side of this vertical carbon sequence, this is the D form. And this particular isomer, the OH is on the left. Here's your vertical line of your carbon chain. Here is the last chiral carbon that's farthest away from your carbonyl. Since it's to the left, this is the L isomer. All the sugars that occur in nature are D sugars. So D-glucose, D-mannose, sucrose, all those sugars have this last OH on the right-hand side. These are all naturally occurring. Glucose, L-glucose, has this last OH on the left-hand side. Glucose, dextrose, is what is known as blood sugar and is the most abundant monosaccharide. Normal blood glucose levels are 70 to 110 milligrams per deciliter. The enzyme insulin regulates blood glucose levels by stimulating the uptake of glucose into tissues or by its conversion into glycogen. Those individuals who have diabetes do not produce enough insulin to adequately regulate the blood sugar levels throughout the day. So they either have to monitor their diet and adjust their sugar levels in their blood by what they eat or inject insulin into their bodies to regulate the body's production, consumption, and regulation of blood glucose. So again, these are all D-isomers. Here is the last chiral carbon farthest away from your carbonyl. This is on the right, it's D-glucose. This is on the right, it's D-galactose. This is on the right, this is D-fructose. So this would be 1, 2, 3, 4, 5, 6. This would be a hexose. It is an aldohexose. The aldehyde. See, the carbonyl is at C1. This is an aldohexose, it's D-glucose. This is 1, 2, 3, 4, 5, 6. This is D-galactose. Notice the difference between here, OH on the right, but here, OH on the left, OH on the right. So just depending on where these OHs are in respect to your vertical carbon chain, these have different names. This is a 1, 2, 3, 4, 5, 6. This is a hexose, but this is a ketohexose. This is fructose. Galactose is one of the components of the disaccharide lactose. Patients with galactosemia lack the enzyme needed to metabolize galactose to break it down into carbon dioxide, water, and energy. The lactose then accumulates and can cause problems such as cataracts and cirrhosis. Fructose is one of the components of the disaccharide sucrose. It is a ketohexose. So here's fructose right here. This is a ketohexose. Keto, carbonyl on the second carbon, 1, 2, 3, 4, 5, 6, ketohexose. It's found in honey and almost twice as sweet as table sugar with the same number of calories per gram. When these molecules are found in biological systems, they are not necessarily in this linear sequence. They are often arranged in a 6-membered or 5-membered ring system. The ring system is formed by the reaction of the OH group and a carbonyl group earlier in the chain. So with glucose, this OH group at C5 cycles around and forms a ring with the C double bond O at C1. So the first step for us to get an understanding of what we're trying to explain to you is to rotate this guy 90 degrees. So this is going to end up on the very far right. This guy is going to go down and end up on the very far left. Or excuse me, 90 degrees. We're going to rotate it clockwise. So 6 is going to rotate up here. 1 is going to rotate down to come off to the right. So all we did was rotate this 90 degrees clockwise. Then the chain has to basically fold in on itself so 1 and the OH on 5 can get spatially close to one another to form a cyclic ring. So 1, 2, 3, 4, 5, and 6, the ends circle around to get close to one another. Here is the C double bond O at C1. There is the OH group at C5. Notice the CH2OH is not involved in the ring formation process. So basically, 1 and 5 have come and grown around toward each other to give us this structure. Then the OH forms a bond with this oxygen, forms a bond with this carbon, giving us this particular isomer. The cyclization can lead to 2 isomers. Since the oxygen that was part of the C double bond O can either end up down or up. So this oxygen attacks that carbon. Carbon-oxygen breaks, becomes a COH. Depending upon how the OH bond is formed, the OH can be down or the OH can be up. Just like the OH is down here and the H is up here. Here the OH is up, the H is down, OH is down, H is up. We can end up with either the OH here being down or up. Where the OH is down, it can be located below the ring. This is known as the alpha isomer, alpha D-glucose. We've created a new chiral center. This carbon was bonded to 3 different things. Now it's bonded to 4 different things. Or the other isomer where the OH ends up in the up position, giving us the beta isomer. So we can form either the alpha D-glucose or the beta D-glucose. These rings drawn in this orientation are called Haworth projections. It's another way to help you get some understanding of the geometric positions of these OH groups around your carbohydrate ring. These monosaccharides don't exist as either beta or alpha. They are in equilibrium with each other. So the reaction is going back and forth. So if you start with the straight chain, we rotate it, we bring these two ends close to one another, we have the non-cyclic D-glucose. If it cyclizes and the OH ends up down, we get to the alpha D-glucose. If it cyclizes and the OH ends up vertically up, we get the beta D-glucose. Turns out that there's a rapid equilibrium occurring between all these systems. They are present, but they're not present in equal amounts. So they're interchanging, but there is one form that is particularly more stable than all the others. So if we look at the mixture, we can see that there's a trace of this open chain system. We see there is some minor percentage of the alpha isomer with the OH group down. The major isomer is the beta isomer with the OH group up. Here is the OH from C5 bonded to the carbonyl. What was the carbonyl carbon? There is the CO from the carbonyl. This OH group ends up. So in this mixture, the beta isomer, where the OH group up, is the predominant form that we find. So starting with the alpha open chain isomer, starting with this open chain isomer, draw the cyclic form, draw the alpha isomer of the cyclic form of D-galactose. So here's our galactose. We're looking at the formation of the oxygen-carbon ring system between that oxygen and that carbon there. So we rotate the molecule around. This guy goes here, that guy goes there. Bring the molecules together. So this CH2OH is still above the 6-membered plane. Here is the oxygen group at C5. Here is carbon of the carbonyl right there. Since we're drawing the alpha isomer, the OH group needs to be down. Then we place in all the other substituents in their relative positions. Since we rotated this down and that up, 4, the OH is up. 3, the OH is up. 2, the OH is down. Other ways that these open chain systems cyclize. Ketohexoses, like fructose, form 5-membered rings with two potential isomers. Again, the OH group at C5 cyclizes and forms a bond with the C double bond O. Here we have the non-cyclic D-fructose isomer. D, the OH here is to the right of our carbon vertical chain. That makes it the D form. Rotate this, CH2OH of C5 is up this way, this rotates down. We have the alpha isomer. This is the alpha, well, let's start here. Here's our open chain. Let's go back. C1, C2. C2 is rotated down, C1 is down. This ends up. These two end up down. Here is C5, CH2OH. There's the oxygen at C5. This forms the 5-membered ring. This ketone can open up and give you the OH down or the OH up. If this OH is down, we have the alpha isomer. If this OH is up, we have the beta isomer. Alpha defructose, beta defructose. These can undergo reactions that we've studied with reactions of alkenes. We can add hydrogen across the C double bond O to give us the corresponding alcohol. So we're reducing the aldehydes with hydrogen on palladium catalysts. So this adds H2 across the C double bond O. We get to this polyol sorbitol. Other reactions that these materials can do. Glucose can be oxidized from the corresponding aldehyde up to the corresponding carboxylic acid. The reagent called Benedict's reagent, it's a copper cation catalyzed reaction. OH is essentially added to this carbon to get you to the corresponding carboxylic acid. Therefore, if we have excessively high levels of glucose in the blood, indicating that someone's enzyme of insulin is not removing glucose from the blood, they can do this kind of test with it, and if they form this brick red color, we know that there is excess glucose in the blood and they have to do something about the blood sugar. Disaccharides are carbohydrates composed of two monosaccharides linked by what is known as a glycosidic linkage. Disaccharide is formed when an OH group of one monosaccharide reacts with the OH group of a second monosaccharide. So here we have glucose starting out. It reacts with another molecule of glucose. This is the alpha isomer. Notice this link is down, but this oxygen at C1 has reacted with the position at C4. These are both alpha isomers. This particular configuration is known as maltose. When the reaction joining these two monosaccharides between two units, the bond that joins these two rings together is called the glycosidic linkage. The glycosidic linkage, the two rings, can be either alpha or beta. Here, we're looking at the position of this oxygen with respect to C1 of this monosaccharide. This is an alpha configuration, therefore this is a 1,4 alpha glycosidic linkage. So the oxygen that is at C1 is in the alpha position. This is a 1,4 C1 C4 alpha glycosidic linkage. You can have disaccharides that are linked in the beta form. The 1,4 here, the OH with respect to C1 of this ring is up. This is a 1,4 beta glycosidic linkage. Hydrolysis of this carbon-oxygen bond breaks it and would form monosaccharides. So if we take the disaccharide maltose, here's our glycosidic linkage. Since this is down, this is going to give us 2 molecules of glucose. This OH group is down, that OH group is down. We have 2 molecules of glucose. So hydrolysis reaction, where we are adding water across the glycosidic linkage, gets us 2 molecules of glucose. We talked about monosaccharides. We talk about polysaccharides. Polysaccharides can join together involving 3 or 4 monosaccharide units joined together. One particular combination of these polysaccharides is cellulose. This is a straight polymeric chain made up of repeating glucose units joined by 1,4 beta glycosidic linkages. Cellulose is found in the cell walls of all plants, where it gives rigidity and support to wood, plant stems, and grass. Think about this. Cellulose is a carbon, oxygen, hydrogen containing compound. Contains no metals whatsoever. But when a storm comes, sometimes buildings that are made of brick and steel don't survive, where those things that contain organic compounds like cellulose, plants, trees, they survive because they have very specific physical properties that other materials, other building materials that we standardly use, brick and stone and steel, don't. So the support and rigidity of cellulose gives it very unique physical properties. There is much research in the building industry to find out how we can use cellulose as new building materials because they have very unique physical properties. Cellulose is the sugar source in plant materials. Animals graze on sugars, or they graze on the plant materials. Ruminating animals, animals that can chew the cud, those that have multiple stomachs like cows, they possess the enzyme that breaks down the 1,4 beta glycosidic linkages in cellulose to give glucose. Why would that be such a boon? Well, what if we could eat grass? What kind of relief would that provide the entire human population in terms of food? Originally, if we look in Genesis 2, God specifically tells Adam and Eve, I have given you every green thing to eat from, plants, seeds, those kinds of things. Man was not initially a meat eater. For some reason, God saw fit to cause the enzyme that was present to break up cellulose into the glucose units for humankind to not have that enzyme. They had it, but at the fall, that was removed from the human system. So humans do not possess the enzyme to hydrolyze cellulose, the beta glycosidase, and therefore cannot digest it. So you can't go out there, pick a clump of grass up or a leaf off or whatever, start munching on it, and get anything that is essentially helpful to you, where animals can. They have the enzymes, humans don't. Cellulose. Here is a structure of cellulose. It is the insoluble fiber in our diets. It is important to add bulk in our diets to eliminate waste. throughout our bodies. So materials like skins of fruits and vegetables, apple skins, potato skins, uh, certain other fruits or vegetables that are peeled. Sometimes those skins themselves can be a helpful dietary product to help us remove waste products from our system. So here we have cellulose. It's the beta 1,4 linkages. Here, this OH group at C1 is up. This OH group at C1 is up. This OH group at C1 is up. This OH group is at C1 is up. So this is the 1,4 beta form. This is what is present in plants, trees, leaves, bark, that provides stability and strength. The enzyme comes in, breaks this off, generates the glucose that's necessary for metabolic processes. Another polysaccharide involving glucose, starch. It's a polymer that's made up of repeating glucose units, not by a beta link, but by an alpha link. So these have alpha glycosidic linkages. Starch is the material that's present in corn, rice, wheat, and potatoes. So with starch, we have this kind of linkage. The C double bond O from C1 is alpha, it's down. This carbon oxygen link is down, it's alpha. Carbon oxygen link is down, is alpha. Carbon oxygen link is down, alpha. There are a couple of kinds of starch. The first kind of starch is a polymer made up of alpha glycosidic 1,4 alpha glycosidic linkages. It is this completely linear chain of carbohydrates. This is called amylose. So amylose is the alpha 1,4 linkage linear chain of glucose. Second kind of starch is what is known as amylopectin. This contains the alpha 1,4 linkages, but at certain points along the way, C6 OH group has cross-linked to another chain of glucose units. So amylopectin has these branch networks. Amylose just has this singular link of glucose units. Notice these are still all alpha 1,4s. The OH group attached to C1 is down. But from one chain, you're connected to another chain at the C6 carbon. Glycogen is a major form of a polysaccharide stored in animals. It's similar in structure to amylopectin. So here's amylose. Here is amylopectin. This is how starch is stored in the human body as essentially fat, such that it can be called upon, used for delivery of energy. In animals, it's the form glycogen. It's similar in structure to amylopectin. Glycogen is stored in the liver and in muscle cells. When glucose is needed for energy, the glycogen or the amylase or the amylopectin is called up from the tissue, and then glucose units are sectioned off there, and used for the energy generation process. Because glycogen is highly branched, there are many ends available for hydrolysis. So here we have glycogen being stored in your liver. So the fatty liver that you have with occasionally due to lack of exercise is just the consumption and storage of too much fat in your liver. Once it's needed for energy, the liver releases it. Since we have branching chains here, we can break there. We can break there, giving us lots of end glucose molecules that we can use to break off and give us energy. Turns out that these carbohydrates are even involved in blood chemistry. There are 4 general blood types: A, B, AB, and O types of blood. A specific blood type is based on 3 or 4 monosaccharides that are attached to a membrane protein of the red blood cells. Each blood type has these monosaccharides associated with them. So D-galactose, L-fucose, and N-acetyl D-glucosamine, these are all the carbohydrates that are associated with determination of which blood type you have: A, B, AB, or O. Type A contains a 4th monosaccharide, not N-acetyl D-glucosamine, but N-acetyl galactosamine. Type B contains an additional D-galactose unit. Type AB has both the components of the A-type and B-type blood carbohydrates. So if we do a very simpler structural diagram, type A blood has N-acetylglucosamine, D-galactose, L-fucose, N-acetyl D-galactosamine. Type B, N-acetyl D-glucosamine, D-galactose, L-fucose, but instead of N-acetyl D-galactosamine, they just have D-galactose. O blood has N-acetyl D-glucosamine, D-galactose, and L-fucose. So here are the O type, A type, B type, and AB type. Here's your red blood cell. Here are the connection points where these carbohydrates attach to the red blood cells that identify them as O type, A type, B type, or AB type. Let me see. So the red one is fucose. This brown one is galactose. The green one is N-acetylglucosamine. That's the O type. A type, fucose, galactose, N-acetylgalactosamine, N-acetylglucosamine, and galactose. B type, fucose, galactose, N-acetylglucosamine, and galactose. AB types. So the AB type has both A components. So here is similar to there. Connected to the red, yellow, blue, green, yellow. Red, yellow, blue, green, yellow. And the B type, red, yellow, yellow, green, yellow. Red, yellow, yellow, green, yellow. The blood of one individual may contain antibodies for another type. Those with the O blood type are called universal donors because people with any other blood type have no antibodies to type O blood. Those that have the AB blood type are universal recipients because their blood contains no antibodies to A, B, or O. That's the lecture on chapter 14. We'll pick up with chapter 15 on Monday. This gets us close to where we need to be with regard to the lecture schedule. I will post something over the weekend on the class page so you can see where we are going to be, what's going to happen Monday, and where we are going to be for the rest of the semester. Have a good weekend. See you guys later. Bye-bye.